So we are already in the middle of January, and from that moment on, the origin of life took place quite relatively soon. So a month later, by middle of February, there's already evidence of life on Earth. And now from this early and promising beginning, nothing happened for about half the history of life on Earth. So we have to go back to middle of July for one of the most important steps in evolution, the origin of the complex cell. Eukaryotic from Greek, with true nucleus cell, with a nucleus and organelles. And from that point on, we're relatively easy in terms of time. By the end of August, we have already evidence for multicellular organisms. And we know that was relatively an easy thing, not only because it happened that soon, but it happened at the very least, there's evidence for 25 different independent pathways that led to the origin of multicellular organisms. By the third week of November, we have complex ecosystems as we know them today. Not because those organisms are still here, but because of the diversity, number of trophic levels, role of predators and prey were already there. So today, I'm going to be focusing on the origin of the eukaryotic cell, arguably the most important major transition in the history of life on Earth, because it unlocked the path towards multicellular organisms like our cells. So welcome to summer 1967, the summer of love, the very same year I was born, and the year where Lynn Margulis, still using her married name, published a landmark paper. After many series of rejections and mocking and patronizing comments, she finally could publish in the Journal of Theoretical Biology a paper that changed radically the way we think about evolution. Instead of focusing on the struggle for coexistence, she proposed this endosymbiotic theory to explain how complex cells arose. And in particular, her point, and there's been many independent scientific support for this idea, originated when a host cell, an archaea cell, phagocytated a smaller cell, a prokaryote, that later on become a mitochondria, and they kind of live in symbiosis. And yet, this still can tell nothing about what compromise, what balance between novelty and conservatism in the underlying mechanisms of protein and gene evolution allow this major reorganization of life on Earth. And actually, it's been puzzling how this happened, because, you know, evolutionary biologists tend to look at intermediate steps, and that led the famous biochemist and science writer Nick Lane to term a black hole at the heart of biology. There are not intermediate steps. You have prokaryotes and fully developed and complex eukaryotic cells. What I'm going to be arguing in this talk is that not only there's nothing mysterious about not finding intermediate steps, but that's the only way that would have been possible. And in particular, as you already mentioned, I'm going to be talking about a paper that my colleagues and I published slightly less than a year ago, in April last year. And that's a paper where we try to provide a perspective about this transition from the lens of physics and computational biology. My three co-authors are all physicists. The first one is a bioinformatician. And in particular, Bartolo and I have been friends for many years ago. We were sharing an office long ago in Barcelona when we were doing our PhDs, and since that moment were spending hours in front of the blackboard in trying to bridge between the problems in biology I was so interested on and the language and perspective of physics. So to some extent, that paper, it's a consequence of these early discussions. If you like slow science, believe me, cannot get slower than that. So,
can you just click on the copy on your laptop? Then I think you're...
Oh yes, sorry about that. Absolutely. Okay. So essentially, the starting point of this research that led to this paper was an empirical pattern. We're making use of a huge database involving about 150 million genes of 33,000 different organisms across the tree of life, and we were just looking at the patterns in gene size distribution. Essentially, this is a log scale, and that's telling you the relative frequency or probability of finding a gene of each one of these lengths. That's a typical example, and that's only for the zebrafish, so that would be all the genes in that particular fish species, but this is a very common pattern across the entire tree of life. So that would be an example of a log normal distribution. So, starting point, just looking at the patterns in empirical data. The good thing about working with physicists is that they are very trained in matching a potential process to explain a pattern.
Can I have a very quick question?
Absolutely.
So, normally genes encode for proteins, for example, but
yes.
A million size gene codes for 1 million residues?
These are all genes. That's a very good question, because all the genes are going to be protein coding genes. That's correct.
But this is a huge protein.
Yes.
Okay. All right.
Yes. It's huge. I agree.
I'm just
huge.
surprised. Yeah.
So I'm going to be mixing in my talk two small tutorials, right? And you are all familiar with that, but I think it's can be useful to remind about that. My first tutorial is just the central limit theorem. And you all know that if you have a set of independent random variables, if you add them, they converge, the sum converges to a normal distribution. So one can just change variables, and instead of having y, we have log of x. Now we have again the sum of these variables. For a property of logs, this is the same as the log of the product. And a derivation of the central limit theorem therefore states that the product of a set of independent random variables, regardless of their initial distribution, are going to map into a log normal distribution. Therefore, because of that, we thought that a multiplicative process in the evolution of genes across time would be a good assumption. And now in the purest tradition in physics, which is using a toy model totally detached from reality and trying to see how far one can go by seeing what are the predictions of this toy model, we thought about the simplest model that encapsulates this multiplicative process.
Can I ask a question?
Yes.
Now you got back to the central limit theorem, so the condition of central limit theorems that you have n independent variable. Now my question is that why the variables themselves are not independent, their logs become independent. Like what changes when you go from the variable itself to the log of it? That only that those meet the conditions of, because the second, the fact that the first and second moment of the distribution is fine for the log and not for the variable itself.
I'm not sure if I understand
there are
your
two
question.
conditions for the central limit theorem.
Mm-hmm.
One is that the variables should be independent.
Correct.
Second is that the first and second moments of the variable should be finite to meet. So probably, but because I'm wondering why the variables themselves, the sum is not meeting the central limit condition, that you only need to take the log to meet the conditions of central limit. Is it because that finally the first and second moment of the log are finite? And in that case was not the case for the variable itself?
No, the variable itself is also finite, I mean, because you have genes like...
But then why the variable itself doesn't follow the normal distribution?
Because it's a log normal distribution.
Yeah.
Okay.
Correct.
So the product
Okay.
of the axis follows a log normal distribution. That's the difference, I think.
Yeah, I guess that means that then the fact that the product, the variables themselves, the sum doesn't follow, it means that they don't meet the conditions of central limit theorem.
No, I don't think so. I think it's the other way around. I mean,
Okay.
when you have a series of variables, if they are independently distributed, right, the product converge to a log normal distribution.
Okay.
So that maybe the question can be turned around.
Yeah.
I mean, your axes are independently distributed. So if you just forget the log transformation, the sum of these axes should actually converge to a normal distribution.
That's my question.
That's what
Does it
the
Yes.
do or
question
not?
is.
Yes.
It does.
I think you should...
Also see,
You
but
derive
that's not
that.
what you are interested. You are on the product.
Correct.
Thank you.
But at any rate, you know, that's an assumption. It could not be the case, right? But again, we're going to be looking at to what extent this assumption is contrasted or validated by real data. So far, if you want, it's a wild assumption, right? We assume that the process that led to this distribution of gene lengths has been essentially a multiplicative process. Therefore, that's the simplest of the models one can think about a multiplicative model of gene growth. Essentially, we start with a series of genes of a given length, randomly distributed. At each point in the simulation, we randomly pick up a gene, and the length of this gene is extended by multiplying original length by a random number that comes from a well-defined probability density function. And the rest of genes are left untouched. So that's the model. You iterate that, and after a large number of time steps, you have a distribution of genes. Now, by doing some algebra on top of this model, you come up with a law of gene growth. Essentially, mean gene length grows exponentially through time according to that distribution, where this is the average mean of the multiplicative factor used in the model. Now, by combining this theoretical prediction that derives by the mere assumption of a multiplicative process with the early on empirical observation of log normal distributions, if you combine these two things, you end up with a predict scaling law that relates mean gene length and variance. And this can be approximated by this simpler equation where you have variance as a function of mean gene length. And that's a typical power law or a scale free, so that if you plot log scales of mean gene length and either the second row moment, the exact equation, or variance, the approximation, right, you have a straight line. You are all familiar with these scale-free distributions. Name this because, you know, as you move across different orders of magnitude, the relationship between the two variables is the same, and that's described by the slope of the curve, which is this exponent here. Perhaps the most well-known of these scale-free distributions is the Gutenberg-Richter law that relates small earthquakes with large ones, right? There's a continuum between the smallest ones to the big one. Therefore, one doesn't need like a different theory to explain small earthquakes than the one to explain the big ones. So far, though, this is a theoretical prediction based on the single assumption of a multiplicative growth process.
Well, what determines here the exponent beta?
Yes.
Is there a theoretical prediction for that, or is it a free parameter?
It's so far it's a free parameter that now you can estimate with empirical data. So the prediction is the power law, it's a free parameter. I'm gonna now be fitting that with empirical data, and that's gonna allow you to fit beta, to find the beta that best fits the data.
I'm just wondering about like what your variance and mean actually apply to. So because you have a distribution of gene length, so if I look at what you're actually saying is if then this distribution changes, the variance scales for
proportional to the mean gene length in that particular way, with that particular relationship. Exactly. That's exactly the point. So far though, again, it's a theoretical assumption, assuming, you know, because of the observation of a log normal, that the underlying process is multiplicative.
You've shown one distribution, so if I would like to test it with data, then how do we test it? Whether can you actually generate multiple points to?
You can just use the data across all the tree of life.
Okay.
Exactly. And that's what I'm gonna be doing that. And that was the first graph that blew my mind, because when you feed data, here you have the empirical points. That's real data on top of this theoretical prediction. And I think there are three take-home messages here, quite powerful ones, I would say. First is that there's a maintained process of gene growth, right, across the entire evolutionary time, from bacteria and archaea all the way to the different groups. Here would be us, you are here, right? And to some extent, this power law acts like a launching pad across which the different groups unfold. Therefore, mean gene length seems to be a good surrogate of organismal complexity. So far, I've been talking about genes, right? But of course, these are protein coding genes, as we were talking. So what I'm going to be looking next is the relationship between gene length and protein length. And when one plots both things, right, mean gene length versus mean protein length, you have this threshold. And this threshold is represented by this equation here. That's the black line. So essentially, and not surprisingly, early on, right, there's a constant relationship 3:1 between gene length and protein length, but there's a moment where this is truncated. It's quite difficult to estimate the point, there's lots of noise, but if now we take only a subset of the data corresponding to completely annotated genomes, we reduce a little bit the noise, and you can see that this is around 1,500 base pairs. From that point on, mean gene length continues growing as we've seen from the previous slide, but there's a tension. Protein doesn't grow anymore, and protein is mean protein length. Some proteins are going to be long, but mean protein length, right, does not change anymore around 500 amino acids. So that illustrates a tension, right, between a constant process of gene growth all the way across the history of life and a change in the way proteins were growing. And of course, because physicists are always looking for like underlying mechanisms and trying to build like more general explanations for that, they already suspected that this could point out, and is a guess so far, the existence of a phase transition. So that was the question we're asking. Is this just a threshold, or could this be an evidence of a phase transition?
I still have a question about your previous slide. So some sort of this seems to indicate that sort of the more complex life has been observed complex life, the more complex the genes are. But some sort of thinking about, for example, like plants. Plants didn't stop evolving the moment they split off from our branch. I mean, from the moment they split off from our branch, there were no flowering plants or whatever. So there has been a whole lot of evolution going on in the family of plants since they split off from animals. But for some reason, this apparently did not lead to more complex genes or longer genes, which is sort of strange because they had their whole evolutionary path and they had their own time going on forming more and more complex genes. So, can you explain this?
Right, it's not the case. I mean, plant genes, as other organisms, continue growing across time. So you see like a maintained growth and a constant growth across time. What doesn't grow, it's protein length.
No, but in the previous slide, if you go one slide, I mean here it seems, one more back, in this one, it seems to indicate that animals have more complex, longer genes at least, than plants. But even though plants also just have been constantly evolving over time since they split off from our branch. So why don't plants have long genes?
I mean, plants have very long genes,
Yeah.
but and they continue evolving, but the mean gene length of some mammals are longer than for many plants.
But is this?
But this doesn't mean they don't continue evolving.
No, but is there some sort of a intuitive reason why complex life should be on the right of your figure and simple life should be at the left of the figure?
I would not say that. I would say the other way around. I would say that mean gene length, because of that correlation, seems to be a good correlate with organismal complexity. For example, what would be organismal complexity? And you can define it. We could argue on the definition, but I would advocate, as many people have done before, that number of different cell types, it's a good indicator of organismal complexity. So indeed, you can see that as you increase number of different cell types, we go from one cell type here, a few cell types, all the way to around 300 in mammals, for example, right? So that aligns with a longer mean gene
Yeah,
length.
but now you're talking about the total number of genes and not the length of...
No, I'm talking average gene length, not total number of genes.
But sorry, I'm just like... so if cell function is diversifying, it means that every cell has a different function.
Right.
But why can't it be that all these different functions are regulated by short genes? I mean, I could have a lot of different short genes, so why do I need long genes to have all these different functions?
Okay, that's, I mean, we're going to spoil a little bit the end of the talk, but the point is that essentially I'm going to be showing that, and of course we are not the first ones, it's very well known, but we provide a different interpretation for that, that the way these genes keep growing so fast in more complex organisms is essentially by having non-coding sequence inserted within protein coding genes. That's the difference. And this is going to allow a much sophisticated process of gene regulation, that's going to explain why you have so many cellular types in mammals, for example. Not because the coding region is longer, but because you have a higher frequency of non-coding, and during splicing, you can have many other combinations.
But by non-coding, you mean regulatory
Introns.
introns.
Yes.
Okay.
And something else, but
Yeah.
introns, mainly introns, exactly.
But that also is the answer to your question, right? I mean,
Yeah.
if you think about vertebrates, our physiology is much more complicated. We regulate our body temperature, for example. We regulate our cell internal environment. So that there is much more regulation going on at a cellular level.
Correct. And regulation is key, but I've not yet reached that. I mean, that's going to be like the way I'll try to wrap that, but that's essentially the key. That's essentially the key.
But so, maybe I'm not, maybe we should ask this question at the end, but if you would remove the introns from the gene length, would that essentially change this picture?
Yes.
Like, okay, yeah.
Totally.
Yeah.
Totally. You would see that gene lengths do not increase.
Because that's the protein length. The coding region is the protein length.
Yeah.
So everything to the right would actually
Correct.
Yeah.
drop down to 1500.
Correct.
So this was also related to my original question about protein length, but okay, yeah.
Yeah,
Yeah.
exactly.
You're right.
Okay.
We'll go back to that at the end. And hopefully I'll be able to wrap things up. Okay.
Just out of curiosity, what is the organism which has the longest average gene length?
I don't remember. I mean, it's not humans. I don't remember that. I cannot tell.
Jellyfish, no?
Is it really?
Yeah, something like that. It's very
Can I ask on the last slide,
The
previous one, here, so, or the one before that anyway, where you see this plateau, this is the mean protein length.
Yes, doesn't
So
mean
what-
that there are.
What happens with the fluctuations? Do they keep growing?
Good point. I don't know. I
Are
don't
they
have.
like
I mean,
some-
I could look at the data, but right now I cannot answer that. I don't increase. Most likely there's an increase in variance as well, but I don't know. Would be easy to look at though. Good. So because could be, again, it's a hypothesis, right? A sign for a phase transition. We're going to be looking at evidence. But before that, as a reminder, allow me to have my second brief tutorial. And this is the Paramount example. You all know that about the second order phase transition, right? Second order, like a Brad, but still continuous. The Ising model, where you have a series of magnets. They can point either up or down, right? You can quantify like +1 or -1. And then you can end up with a macroscopic parameter, what physicists like to call an order parameter, which is the absolute value of the sum of -1s and 1s. And essentially the idea is that you have this parameter you can fine-tune, which is the temperature of the system of thermal disorder, right? When temperature is very high, right? Essentially randomness dominates. And the deterministic part, because these magnets tend to align, you tend to point up if all your nervos are pointing up, but randomness dominates, each magnet is either up or down randomly, and therefore, you know, this order parameter is 0, right? Because that you have on average as many positive ones than negative ones, and then it gives 0, 0, 0 up to a point, a critical temperature, where now it becomes positive because the deterministic part starts dominating, and at the very end there's no randomness, right? So all magnets point either up or either down, so order parameter is the maximum, the size of the magnets, the number of magnets. So that's the typical example of an order parameter. 0 before the critical point, positive after that. That signals a critical temperature, and there's lots of statistical properties of this critical temperature. For example, one can define a correlation length that tells you like the distance up to which one variable affects another one, and essentially near the critical point there's a power law relating how this correlation length scales as you are getting closer to the critical temperature. And that would be a class of universality, because that critical exponent doesn't have like multiple values, but they do have a value. In the case of percolation like this one, because the critical point is also known as percolation point, where you have clusters of all sizes from just one little site to a cluster spanning the entire system. That's another way of saying that the system percolates and that exponent is 1. Okay, that's only a reminder, but it's going to be useful because now we're trying to come up with an equivalent order parameter. And our order parameter, and we are getting back to what we were discussing, right? It's the fraction of non-coding sequences within a gene. That's what I'm plotting here. Fraction of non-coding sequences, right? And again, this is, as always, mean gene length. So this is the data, and black would be that expression here, where fraction of non-coding sequences is 0 all the way to this point, and from that point on gets positive and keeps growing. Again, see humans. Pretty much 90-something% of the length is occupied by non-coding sequences. So that would be an equivalent of an order parameter that seems to suggest that's indeed a second order phase transitions, but we can do better than that.
So, does this imply that bacteria don't have gene regulation?
No, not at all. It just implies that they have the original way of regulating, which is protein-based. And I'll go back to that, but just to summarize that,
So it's
essentially,
transcription-based.
correct. Essentially, early on in the history of life, there was only one way to regulate gene activity, based on proteins. And then life could become more sophisticated by elongating proteins.
Genes.
No, proteins.
And
Because
proteins.
proteins
It's exactly
proteins is what controls gene regulation. So you have longer proteins, and you can do more things. You have more degrees of freedom. You can be more. But as the point at the end is that this can be done up to a point. And then we'll introduce the beautiful idea of constraints and what kind of constraint this is, right? But again, we can do a little bit better because there are theoretical predictions, statistical predictions, or properties of second order phase transitions. I'm going to be using 2 now, and later on a third one. The first one is the idea of critical slowing down. And essentially that means that as you're getting closer to a tipping point, it takes longer for the system to settle into its attractor. Meaning that there are multiple solutions for that particular value of the order parameter. And we can now look at data, and what we are plotting here is for a given mean gene length, what's the number of solutions, and solutions means what's the number of values of fraction of non-coding sequences. And for many species, this is pretty much, there's no variability, but number of solutions increases near the critical point as predicted by critical slowing down. There's a larger number of solutions near the critical point.
Sorry, I didn't follow exactly what are these states? Is the
These states would be like for a given mean gene length,
yeah.
you have all organisms, right, that have this mean gene length, and then you see the variance in the fraction of non-coding sequence.
So this is the variance.
Yes.
Okay.
Okay, it's variance. Okay.
Yes.
Yeah, I can't read what's on the vertical. It's a fraction of
States.
states.
Fraction of states.
But it's
Yes.
actually the variance.
Yes. Fraction of states would be how many different states, how many different values of fraction of non-coding sequences for that particular mean gene length. So species that are around this critical mean gene length, each one may have a different value of fraction of non-coding sequences.
It's like it maximizes something like entropy on that value of
Correct.
gene length.
Correct. You could think in those terms.
I don't know if it's entropy.
It maximizes the number of solutions.
Yeah.
Why is that? Because there are more metastable solutions. Why is that? Because it takes much longer for the system to settle into the stable solution.
Yeah, because there are so many options.
Correct. There are so many options.
Usually this is interpreted as degeneracy or lower driving force at the critical point. So for example, there's no surface tension in the Ising model.
Yeah.
So is this something similar? There's no...
It's absolutely the same.
Okay.
It's absolutely the same. It's another way to phrase it.
Okay.
It's absolutely the same. You have less tension,
Yes.
therefore it takes longer. Like the potential surface has a lower slope, it takes longer, and therefore you can end up finding different values for a given moment in time. You could see it like that.
So my background is mostly physics, so I'm just looking at this very interesting plot, and I would have actually interpreted this more like a percolation type phenomenon.
Correct.
Than nothing to do with the easing model. Right?
Correct. It's percolation.
Okay.
It's percolation.
He also said it.
Oh, yes.
No, but I'll give you another evidence.
Okay.
You know, that's one property of second order phase transitions, and again, it seems coherent with data. The second one,
But
it's...
sorry, Jordi. I mean, how do you define these states? Fraction of states?
Yes. Again, let's imagine that for each window of mean gene length, you may have a few thousand different organisms that they all have this mean gene length, right? And now you look at each one of them and look what's the fraction of non-coding sequences. It could be 5%, it could be 20%, it could be 45%. So you are plotting the number, if you want, of different states or the variance.
But then how does that relate to the fact that you actually have this power law invariance in your gene length versus the mean gene length? Because you also said that the coding fraction, the coding length doesn't change. That's around 1500. So I expect then that the variance for larger gene lengths will actually grow, because that's what your power law says. So I don't understand why the curve actually goes down in this last...
Right, because you could have an increase in variance, right? But you could also have that all the species with that particular mean gene length, they all map to the same fraction of non-coding sequences. I mean, it's what you observe.
Okay.
It's what you observe. But what you're pointing out is a this tension, and I think that's a key word of that. There's a tension between two different forces. On one hand, you have a constant process of gene growth, early on in the history of life, that goes hand by hand with how proteins increase, but at some point there's a discontinuity. And the nature of this tension is not informational. We think it's a hypothesis. You may disagree with that, but...
I just have a question about how I should interpret the previous plot. If you go back one slide. So at the beginning of your talk, you showed a plot where, for a given organism, there's a log normal distribution of gene lengths, right?
Correct.
Each one of these genes will have a particular fraction of non-coding sequence.
Yeah.
Right?
Well, the log normal, no, the log normal just...
In a given organism, shall we
Yes.
say, right? So if I look at this plot, I see dots that seem to correspond to different types of organisms. So
Correct.
Why wouldn't they scatter more? Like just, do you see what I mean? Because every organism can have long gene sequences, right? Of which...
But up to a point, you remember that in the power law, you see that the different monophyletic groups unfold in the evolutionary order across that power law. So the first class belongs to bacteria and archaea, then you have fungi, then you have plants, then you have invertebrates, then you have vertebrates.
Could I have done this same thing for just a given organism? right? So I take a single organism and I just look at the mean gene length and look at how much of the fractions are non-coding sequences. Would I also get a plot like this for a single organism?
I don't think so, or at least you would have much lower variance, because in a single organism, essentially, terms of gene regulation are the same. Where it is in this continuous between the tension of the two processes is going to be the same, so you would not find that level of variance. That level of variance can only be observed across a wide range of organisms for the reasons that will be apparent later on, which is at some point there's a shift in how you regulate gene activity.
And shouldn't the variance, I mean, it should be, so before a tipping point, the critical slowing down occurs, right? Before a phase transition. But once the phase transition is there, then it should go down, right?
Exactly, that's the reason it goes down, the number of states. Exactly. So that's the typical behavior of second order phase transition. Critical slowing down increases, time that the system requires to settle increases before the critical point, and then
so the
it
peak
decreases
is
again.
the critical point.
Exactly. The peak, its line corresponds to the critical point. Exactly. And by the way, for the ones of you interested in ecology, there's some of you that have been working on that. That's the very same unit or concept that is used by the work that I've done with my good friend Martin Schefa on early warning signals. It just builds on this very concept of critical slowing down.
It looks from your plot that fungi are critical.
Yes.
What does that tell us about fungi?
Yes, that's a very good point. Fungi are critical, and a few other organisms, but in particular fungi. But you can see the same with organisms, that across evolution, they at some point become towards a simpler level of organization. And that typically is the case when you have parasitic life forms. That after kind of inventing across evolution the parasitic way of life, they tend to become simpler, because essentially you don't need all this repertoire of genes and proteins and regulation. So you have a little bit organisms that go back and more variability in this. Yes. Okay, so the second evidence for a phase transition is the following one. You can derive, right, a short mean gene length as this, that's similar to the Ising model, essentially average gene length minus critical gene length over the critical, and then once you define that short mean gene length, you can show that the critical or the order parameter, again the fraction of non-coding sequence, above and close to the critical point follows this scaling law, and the exponent is equal than 1 as in percolation. I think you were making the point. So indeed, that seems to suggest that this is the same broad class of processes that take place in percolation.
I have only one question. In terms of the physical picture, is percolation of what quantity in this case?
That's hard to tell. I cannot really explain. We've discussed about that, and Bartolo had an interpretation, but I have to confess, I found it a little bit far-fetched. But you could think in terms of percolation, once you move from a network of gene regulation to a network of networks. Essentially, when you have different organisms living in the same.
There are clusters of
correct.
organisms, and they.
So then you have this problem of networks that interact, and you could define percolation in these terms. But I have to confess that I found it difficult to.
But there is
follow.
some underlying self-similarity. That's why you get power law. So it's somehow maybe not calculated. I don't know. I'm just trying to get some intuition.
No, that's good. That's good. All right. So far two evidence, I would say serious evidences, right, of phase transition. So let's assume that's indeed a phase transition. The next question is, what type of phase transition? And here is where things become more interesting, and we are now moving into trying to apply some of the points, intuitions that you already manifested early on, right? And just think about protein growth and the constraints in protein growth and the type of unit, because at the end of the day, information is key here. We're encoding information in gene sequences, we're encoding information in amino acid sequences. And the point, and this is again something that has been advocated and proved by many others empirically, finding a protein of a given length becomes more and more difficult as the length increases. Why is that? Because proteins are not linear, they are linear sequence, but the key point is their three-dimensional structure, so proteins have to fold. And now it's very well studied that the time it requires for the folding of a protein grows very fast, probably exponentially, I don't know for sure that, but it grows very fast with the length of the protein. And if it takes too long, essentially the protein decays. And now this seems to be, or this is typical from this kind of searching algorithms, for example, in the problem of the salesman traveling or coloring maps, right, in the so-called NP-complete, non-deterministic polynomial time complete problems. In these kind of problems, finding a solution becomes very, very expensive, time-consuming as the number of degrees of freedom increase, but once somebody presents a solution, it's easy to determine whether it's correct or not. That's very easy, right? Polynomial time, it can be done in polynomial time, but finding the solution seems to grow very, very hard with the size of the problem, right? We're going to be advocating that evolution is a sort of searching algorithm. Now you may think this is a little bit fetched out of reality, but just remember that a very well-known class of algorithms is called genetic algorithms, right? And even more than that, a few years ago, 2 or 3, I forgot, it may be 4, not more than that, Nobel Prize in Chemistry was given to people who had used evolution through natural selection as a way to come up with new chemical molecules. So this idea of evolution as an algorithm is by no means something that we suggest to begin, right? So, in order to think about algorithmic transitions, let's play Sudokus. Essentially, when you have this, it's a simplification of Sudoku, you all know you have 1 to 4, and there are constraints. You cannot have twice the same digits in a column, in a row, or in a block, right? In this case, we have high number of constraints, small number of solutions. Number of constraints and number of solutions are the two sides of the same coin. When you have that extreme case, where number of constraints is so high, it's extremely fast, it's extremely easy to find a solution. Like, for example, this one. Or, it's extremely fast, it's extremely easy to determine there's no such solution. There's no solution because you would violate the constraints or not repeating the same digit in either a row or column. Now let's go to the other extreme. Let's go to the extreme where now we've been increasing number of solutions and decreasing number of constraints, right? Number of constraints is very, very little. You can play with all numbers except having that digit here, right? Now you have an increasingly large number of solutions. So now you shift finding the solution by finding a solution. It's very easy. That's a solution. If you don't like it, as a famous quote by Groucho Marx about his principles. If you don't like them, I have others. Here you have another solution, and yet another solution. You see, the number of constraints is very, very low. Number of solutions is very high. So it's relatively fast to come up with one solution or another one. In the middle, though, we have an intermediate case with number of constraints have been decreasing, number of solutions been increasing, and that's more difficult. Essentially, what you have, if we put all this together, right, is that there's the so-called easy, hard, easy pattern in searching algorithms. Early on, right, algorithmic complexity, which is the time it takes for you or for a computer to solve the Sudoku, keeps growing as number of solution increases, reaches a peak in hardness, and then it decreases as time goes by, because essentially the algorithm changed its strategy. Instead of trying to find the solution, it just comes up with a solution. And what happens now is that complexity decreases as number of solutions keeps increasing. That's pointed out by a phase transition, and in this case the phase transition would be in the amount, what's the fraction of randomly generated seeds that are solvable. Randomly generated is you generate like the template for a Sudoku here, and you do it like 1,000 times. What's the fraction that can be solvable? And early on, that's pretty much zero, because there are so many constraints that if you do randomly, you cannot just find a solution, right? But at some point, you know, it becomes almost one. That's an example of a phase transition, but now of a computational, of an algorithmic nature. So that's a good framework to think about these phase transitions, because information is key, and because we assume that for evolution it takes some time to find a solution.
Just to understand this plot, is the complexity of the optimal algorithm?
No, the complexity here is time, algorithmic time. Perhaps it's easier.
For instance, you could imagine also using random search at the left end. That would be very horrendously inefficient, because almost every solution would be wrong. So, which algorithm are you using to define the...
Let's think that regardless of the algorithm, if you have the same algorithm, it's gonna take more time to find a solution.
Okay.
Could be a randomly, one that randomly explores all possibilities. I think that's the standard one, right? For that complex algorithm, if we do not change the algorithm, you always will find that there's an increase and then a decrease in the time that requires the algorithm to solve that solution.
So, I mean, this seems to suggest that exactly at a half, where the number of constraints equals the number of solutions, the complexity is highest. Is that always the case, or is it just an example?
That's just a made example. Actually, that doesn't represent a system. But the key point is that trade-off. I don't know if it's exactly half and half. This I cannot tell. That may be system dependent.
Yeah, sure.
But what's general is that you have this peak and then a decrease. Yeah, it doesn't need to be exactly half and half. But the key is that, the reason for that is this trade-off between number of solutions and number of constraints.
So going back to the question, I think another way to phrase it is how aware is the algorithm of the constraints? Because in the random search process, it's ignoring any of the constraints. So there is this question about the availability of the information of the constraints. So how is that factored in? And I guess in the real search process, there is some information about the constraints in the search process.
Yeah.
Right?
I don't know. Actually, that's a very good point. I guess I would say there's no need to go into that. We could assume that there's no information on number of constraints. If you assume there's a given algorithm that works one way or another, and one way could be like trying to randomly brute force solving the system, you have an increase in time as number of constraints decrease and number of solutions increases, and at some point that decreases. And that would be very, very general, regardless of whether or
So
not.
the shape may change, but
Yes,
it's still...
exactly. The shape is going to change. It's going to be context dependent, right? But the statistical properties or the easy pattern next to a phase transition of a computational origin, it's always going to be the same. If you have one phase transition of this type, of course. But that happens, for example, in all the NP-complete problems. You name it, you'll find that. Of course, that's going to be a little bit towards the left or towards the right, but you'll have something qualitatively like that, which can be associated to a second-order phase transition, and you have a peak. It can be asymmetric, it can be more towards the right, but the qualitative properties are going to be the same. This play between constraints, number of solutions is going to be the same. So going back now, that's a little bit of an analogy, right? Going back now to the problem of proteins, right? So what's algorithmic complexity? As before, it would be the average time spent per size unit to reach a solution. That's the definition. Now, in our case, time spent by evolution per nucleotide, right, to reach a species characterized by a given mean gen length. That's essentially the definition of algorithmic complexity. The only thing I'm going to be doing now is trying to apply that to the problem of evolution, and therefore trying to estimate those parameters in an evolutionary setting, right? But before that, let's go back to this graph, right? You remember that was critical slowing down that I was showing when I was showing the order parameter, because on top of this graph, now we are going to be building the empirical estimate of algorithmic complexity. And in particular, how are we going to be doing that? Well, algorithmic complexity, or time needed to find a solution, is going to be estimated. That's our way of doing things with the constraints of the data we have, of course, by dividing divergence times for different phyletic groups, using humans as a reference, right, divergence time, over average mean gene length. That's the way to estimate that equation of algorithmic complexity. Now we have the data, we can estimate that. And when you do that, right, this is the results. These are the results. Each point corresponds to a different monophyletic group. Again, from completely different perspectives, right, completely different ways of doing that, there's a clear overlap on the peak, again, close to the critical point. But now we can do a little bit better in terms of going deeper into not qualitative predictions, but quantitative predictions. Essentially, let's assume that, indeed, this is an algorithmic phase transition. Again, you will say, you've not proved that, and I will say, you're totally right. But again, very common way of thinking for physicists. We assume that's an algorithmic phase transition. What would be derived from that? And then we can go back to the data. Well, if you assume that's an algorithmic phase transition, by definition of a phase transition, you'll be finding a peak of hardness, right, at the critical point. Essentially, that means that the derivative of algorithmic complexity over average mean length at the critical point is 0, right? Yes. So, by doing that assumption, and going back to data, you can solve using the loss of gene growth. I show you later on how mean gene length grows through time exponentially, right? And then, of course, there were a couple of parameters. You can try to do the best you can, lots of noise, absolutely, but that's what we have, right? And by fitting data, right, for example, original gene length is estimated by the data in the scaling law, right? You estimate that point, and then the estimation, assuming this is indeed an algorithmic phase transition, is that the critical length is around 1,500 base pairs as observed in data. But you could also solve not by critical length, but critical time. What's the moment in time in evolution where that phase transition took place? If you do that, right, you have just this parameter, which is the, I'll remember, it's the mean of this multiplicative process, which is at the key of the multiplicative model of gene growth, right? Again, you can estimate through the fit of the law of gene growth with the data. That gives an estimation of 2,600 million years ago. Now, we can now try to compare that estimation with the best and more timely estimation of the moment in time where the eukaryotic cell arises. So that would correspond to the first eukaryotic common ancestor, 2,900 years ago. That paper, completely independent group of people, by combining both molecular data and paleontological fossil data, right? That's the moment where you have the first eukaryotic common ancestor. That's the moment, the estimation, where you have the last eukaryotic common ancestor, and everybody derives from that point between 2,300, 2,900 years ago, quite very much in the range of our estimation. And with that, I'm going to be finishing this set of evidences, and I'll try to wrap it up and go back to some of the questions you were making. So now, why these great physical results, analytical work simulations? What can we learn? Going back to biology, right? I'm the biologist on the team. Is there anything that is telling us about the way evolution works? How to understand those results? Now we need to, in this dialectical process, we need to add some biology, right? And to me, that boils down to the role of constraints in evolution. I've been a big fan of the role of constraints, very much inspired by my former PhD supervisor, Per Albert, developmental biologist Per Albert, who worked on this idea of developmental constraints and how developmental constraints are key in understanding evolution, because as he would say, yeah, natural selection picks up who is the winner of a game, but developmental constraints is telling who are the players to begin with, right? So in this case, and trying to wrap this up, we have the following picture. And again, that's a speculation, right? Early on in the history of life, we're going back to what we were talking with André, we have that there's a mechanism of gene regulation based on proteins. Proteins, some proteins, are used to regulate genes, the activity of genes, when a gene is on and off and so on, right? And of course there's been evolution across since the beginning, but this evolution was following the only path, the path of using proteins as the regulator. And things could become more interesting, could become more complex within the domain of organismal complexity of a prokaryotic cell by elongating proteins, right? So proteins can be elongated parallel to gene, hand by hand, with the way genes were increasing. You can do things more complicated by having longer proteins. But I would argue evolution reached a wall. That's a constraint. And it's a constraint of an algorithmic nature. You cannot just make proteins longer at that point. That's a physical impossibility. And I think that explains, it's my elucubration of course, why nothing happened during half of the history of life on Earth. Until, if you like to believe in my records, that's the closer one, the origin of the eukaryotic cell through this endosymbiontic theory. That's very, very unexpected thing. When that happened, lots of cascading consequences happen. One of them is that the original prokaryote that become mitochondria, living in a very stable environment, didn't need all the genes it had. And having genes is very costly metabolically. Producing proteins is very costly, right? So evolution tends not to do things that are not necessary. These genes were released, and many of those, not all, but many of those, become introns, reach the nucleus, were abused by the gene of the archaean cell. And now suddenly what happens is that evolution was able to sort up this wall, this computational wall, by coming up with a novel mechanism of gene regulation. We're going to be going from a linear model of gene regulation to a combinatorial one. Because suddenly you have these introns, what people thought trash DNA, things that serve nothing, right? But become very, very important. Because in the process of transcription from genes to messenger RNA, which happen in the nucleus, right? Those introns are also transcribed. But before this mitochondrial RNA leaves the nucleus, they have to be removed, right? And this is a sort of molecular scissor that takes away the introns from messenger RNA. And now you have all these different blocks. You have a Lego, if you want, with multiple pieces, and you can play. Any of you can play in different ways. So you can combine all these things. You see, so suddenly you have this combinatorial way of exploring things, and therefore you have a much faster way of regulating genes. And suddenly you can go into the difficulties of regulating a network of networks of gene regulation. What I think is so beautiful about this work, and my own take-home message, is that going back to these early times, right, in the early 90s actually, of last century, sounds like, oh, okay, we're getting on. Where Bartolo and I tried to discuss on the blackboard, that's a homage to those times because it helps to reconcile the stochasticity and irreproducibility of evolution, right? Nobody could have, there's no physical theory that would have predicted 2,000 million years after origin of life there's going to be this symbiosis. That's, if you rewind the history of life, as the famous experiment of Stephen Jay Gould, you run it again 1,000 times, perhaps we never come up with that. And, you know, life would be just bacteria. That's biology, that's history, that's stochasticity. And yet, once this happened, things could not follow from different points of view. There's only one way where things could follow. That's where the universality of physics, the predictive power of physics comes up. And to me that's mind-blowing. To me that's really, really inspiring. This dialogue between biology and physics, and this ability to go back to predictions. And no one has said it as beautifully as the late Nobel Prize winner, Gene Wigner. I don't know if any of you have read this paper. You've read this paper. Very good for you too. Okay, a few of you. For the rest, if anything comes out of my talk, you can forget everything I said. Read the paper. It's one of the most beautiful papers ever written. And I'm going to be ending up with a quotation from that very same paper that reads, "The miracle of the appropriateness of the language of mathematics for the formulation of the laws of physics," you can add biology here as well, "is a wonderful gift which we neither understand nor deserve." And I'll stop here. I not forgot that.
Yeah, I have a question about your previous slide, because while you were talking, you said, okay, then I can build I have Lego blocks and I can build things. But now I'm confused about what do you call a gene and what do you call a protein? Because this almost seems like from one gene you can have 3 different proteins.
That's the point. That's exactly the point. From one gene, for a finite length of a gene, now you have multiple proteins. And therefore, you have multiple elements in the novel process, in the new process of gene regulation.
So if we now would look at, so what would this imply for the number of proteins versus the number of genes or the gene length? Because this is, this would suggest, given that the protein length stays constant, that in fact the number of proteins grows in species.
Yes. You have more proteins.
Because I understood the introns
Exactly.
actually as more as...
That's another way of saying.
As
So
regulatory.
you are shifting from having a protein of an increased length or a finite number of proteins or a given number of proteins of an increasing length to having for an average fixed length different types of proteins. And not only proteins, because some of these become RNA, not necessarily protein, but RNA now plays a key role in gene regulation. So it's proteins and other things, but they all derive from the process of having introns and the necessity to cut those introns. And that only happens when you have the merge of two different organisms and a network of networks of gene regulation. And there's many other things that of course I cannot talk, but the point by Nick Lane, wonderful work by him and his colleague, I forgot now, Bill Martin, they claim that once you have that way, you cannot just have like, okay, eukaryotes that have the gene regulation but not nucleus. And everything becomes a necessity. Why is that? Well, you have two time scales. You have a very fast process of transcription of DNA into RNA. But then this goes to the cytoplasm and is traduced into proteins, right? So it would be terrible that the messenger RNA, without removing the introns, goes there, because there's going to be lots of problems, lots of proteins, and therefore you have a nucleus, according to them, that's their interpretation, which is like a way of buffering these two processes of different time scales. You allow enough time for the scissors to operate, right? Splicing, alternative splicing to operate before they go out and then can be traduced into proteins.
Similar to this whole discussion, is now the origin of this whole framework the process of endosymbiosis, or is the symbiosis a consequence of this mechanism?
The other way around. Endosymbiosis, and that's a little bit of a speculation, right? This part, we're in the discussion part, right? The way I would think about that is that endosymbiosis create a new scenario, and then that's the follow-up or the consequence of the endosymbiosis.
Yeah. Okay. So
because before endosymbiosis, you don't have that. Technically is not correct. You have some introns that are from a different nature, like viruses, but let's put aside, before endosymbiosis, you don't have this huge number of introns.
No, but okay, but I understand that symbiosis is the origin or the cause of this effect. But then I don't understand your phase transition stochasticity argument about the timescales, right? Because if that's the origin, why do your theory actually predict this 26 million years? Because it all depends on something completely different, namely the endosymbiosis.
Correct, but what the theory thinks, that's going to help to fix things. I understand that was a little bit confusing from my side. The theory of an algorithmic phase transition and all the analytical properties derived from that allow you to make a prediction of that time, and that actually fits the moment estimated by different means where the endosymbiotic event happened.
But they said that completely unlikely. Okay, so maybe we should discuss this over lunch, because I,
No.
I find it absolutely unlikely, right? I mean,
But what's unlikely that this endosymbiotic thing happen, right?
Yeah, but also that you can predict that right on track, because it's as you said, a stochastic process, right? So how can you? It's only the average which you expect to be.
I don't understand. I don't deserve.
Okay, you might be lucky.
No. Let's go back. I think that's an interesting point. Your claim would be that it's very unlikely you can make those predictions, and they fit.
Well, it would be likely to actually do the average time you actually have to wait.
Right.
But I mean, the history only happened once, right? So I mean, how likely is it that you're right in the right spot? I mean, if you do it's like throwing a dice and you exactly, well, maybe not a dice, maybe many dice together, and you exactly get all the sixes.
I think he was a couple of 100 million years out. So.
Yeah.
Maybe that's it.
I mean, there's some variability, but the point, you're totally right, that happens only once. So we cannot use about ideas of whether we have like different planets with similar evolution, how often would this place, although in the reggae would say very unlikely. You would say, why you say that? You have only one. You are correct, but two things. Why it took so long? That to me signals it's very, very unlikely, and why it only happens one. On the other hand, the origin of multicellular, to me it's easy. If you have life, unicellular life in other planets, I would say, and it's a bold claim, of course, I would say, just wait enough, you'll have multicellular life. What you've been smoking, how you make that point? Well, because first it took relatively very little, and it happened at the very least 25 times. That suggests to me it's easy. It's easy. Right.
But it's also a kind of a post-diction, right?
Yeah, so
No, that's right, but I don't
know
think...
from knowing now, going backwards, that something must have happened at that
Exactly.
time.
So I think your point of this analogy is a little bit misled, because it's not that you predict when something should have happened and it's just there. It's the other way around. I mean, something happened across evolution and left a molecular signature. And now the point of the paper is, with this macroscopic analysis, those basic statistical distributions, you pick up that signal. And then you can just go back to the moment it happened. But it's not like I predict this miracle is going to happen and it happened. It's the other way around. You have a history. It could have happened 1,000 million years ago earlier or later. It doesn't matter. But that would have left a signal, and then we would be able to trace the moment where this happened. It's the other way around.
So I do have a question about the last slide, one previous one here. Well, first, just I think that the idea of viewing this as sort of an algorithmic complexity problem is very interesting, and that you see the difference. You can almost think of these two different sides as sort of a different algorithm to solve a problem, and encoding for proteins in some way, and then that transition is sort of, if I bring the parallel to Sudoku, right? So on the left side, you are thinking about just finding a valid solution, and on the right side, you're thinking about any solution. So there's a difference in strategy of your algorithm, and this is also kind of a difference in strategy. But I remember from the beginning of your talk that your model is the left side, the multiplicative process, taking a gene.
Both sides. Because remember, the process of gene length has been constant across the history of life, with at the very least the same average multiplicative process. So both.
Okay.
Because remember, that was the power law. That was the power law. When we were only looking at genes, the power law is telling you that whatever happened, whatever model was the most appropriate one, that's been maintain. You don't have a change in the slope, right? The difference happen here, and that corresponds to the region of the eukaryotic cell, because there's been a tension between that constant process and that process early on parallel, likewise similar, of protein length, until reaching a wall. And that's telling you why you saw prokaryotes for so long. You see nothing else, because evolution hit a wall.
And why does the algorithmic complexity decrease once? I mean, I did not understand that, but I think like you showed this graph where you plotted the algorithmic complexity on top of the variance.
Right. Essentially,
So what is
because
the explanation there?
yes, I'll try to give you like two levels of explanation. First, the computer science explanation, and then the analogy in biology. In computer science, with problems as the salesman traveling and things like that, it's just because when the number of constraints.
Yeah, that part I understood, like the Sudoku
Okay.
example.
Yes.
But what is the biological intuition for that? Like what? How do you explain that in this?
I think a little bit like you were saying, right? Like here you have the strategy here is finding the solution with mechanism of finding that with a constraint, because the mechanism is only like a linear way, right? And here you remove that constraint by changing the nature of the game. You are opening to a different way of regulating. And once this happens, and that happened because a stochastic evolutionary event, right? Once this happens, then you have many more solutions. And therefore the task of evolution is not finding the solution that somebody has in mind. Evolution doesn't care, it's blind, right? The role of evolution is finding a solution. When you have a huge number of solutions, perhaps finding the solution, if you and I decide that this is the solution we want, it will be as difficult, or even more difficult. But this is not what the point is. The point is that finding a solution.
And is that true that in the right side, the same protein is encoded in many different ways? That's something that you would be able to find in the data, right? So,
okay, let's
if
see.
the
In the
solution
right
is
hand side, the same.
encoding if the solution is encoding a protein,
Yes.
and you're saying that on the right side, it's more about there are many different ways to find a solution, then you would expect that the same protein would be able to be encoded in many different ways by combining different blocks.
That's correct. It's a bit more complicated because, again, it's not only that you continue regulating only with proteins. Suddenly now there's a new toolkit of gene regulation, and I'm not an expert. I actually know very little about that. But essentially, you have microRNAs, for example, that also play a key role, and this has been the material of several Nobel Prize winners in the last recent few decades, that they discovered that these things play a major role in regulation.
So,
Okay, sorry, I don't know who was first. Sorry about that.
Yeah. So maybe my timeline of evolution is not complete in my mind, but so what you're saying is on the right side of the phase shift, we have this phase transition, we have this complexity, and this complexity comes from the fact that we have introns. So for some reasons, on the left of the transition, we don't have introns. And so you're saying essentially the fact that we are eukaryotic cells and we have this nucleus, this helps in some sort of protecting or the introns, or making at least introns useful. So are you not just arguing that the existence of the nucleus is the important driving fact and not necessarily endosymbiosis?
No, well, it's a matter. I mean, at the end of the day, is you know, being more relaxed, right? So it's not only one thing, but a combination of things. But I think the way I would say is that endosymbiosis is the most important point, because everything derives from that. You still need other things, of course.
But
it's not too
what it means in the case of bacteria, I mean, bacteria are constantly exchanging DNA on this horizontal DNA transfer and so on. So it would be for bacteria very easy to get introns, just random pieces of DNA.
It seems that's not the case, and that's a reality. I think it would be wrong to say there's absolutely no introns in bacteria. I think technically that's wrong. But if you look at the empirical fraction of non-coding sequences, yep, here it is. I mean, you see, there are exceptions, but these are already proteins. I think for bacteria, yeah, there are a few. You're totally right, but very, very minimum.
But what would be interesting for me is that there apparently is an evolutionary mechanism preventing bacteria from having introns in their DNA. And I'm not sure where that would come from in your story.
Well, that's a great point. First, two things. My story is very limited. I try to make sense of something, and there are many things that this cannot explain, of course, right? But the second point is, I would not say that. I would not. It seems that your reasoning is that what mechanisms prevent bacteria from having introns? You may be right, but I would say I found more parsimonious the alternative way of thinking, that bacteria did not get enough introns for these kind of things happening, because the measured income of introns is after the endosymbiosis, because it's with many genes of mitochondria that are released, and that's huge. That's much faster and important than other process of horizontal gene transfer. So that's the way I would explain. But of course, I mean, here we are speculating. But I think it's important. At the very least, it's clear that when the endosymbiosis happen, multiple molecular evidence would be using Occam's razor would be almost impossible to explain, like what happens across different lineage, and the similarities in mitochondria, the similarities in chloroplast. That would be impossible, right? So that's correct. And then it's also correct that the original prokaryotes that now become mitochondria, also chloroplast in plants and others, but mainly mitochondria, had many, many more genes, and those genes were released, and that's the raw material for introns and what unlocks this other way of regulating things. But you are totally right. You may not buy that. I cannot prove that. We can prove all these things. You know, you have the data, you have the theory. You can disprove me if we make a mistake. But that's theory. That's data. You're totally right that in the wrap up, in the discussion, this is an interpretation, and it may not be the most solid ones. It's just the one I think fits better the story.
Yeah, so I think very great talk. To go back to computational systems, so the genetic algorithms you mentioned, is it safe to say that genetic algorithms currently are still on the left side of your curve?
Current genetic algorithms
are in this black hole of because the genetic algorithm didn't make any big progress in the last 20 years.
No.
Is there something we can learn in building genetic algorithms?
No, I think here there are two different. We're talking about different things. I would say no, genetic algorithms are not in the left-hand side. I would say genetic algorithms. Let's see if I can get a little bit faster. genetic algorithms are both in the right-hand side and the left-hand side, right? Because that would be the same algorithm, the same genetic algorithm, the same, you name it, algorithm, right? It's here and it's here. What changes is the shift in how the genetic algorithm finds the solution. Because early on, there's only a solution, at some point there's multiple solutions. So I think that's constant. What my story, or what the biology that we try to look through this lens is that it explains why you have more solutions in that site.
Great talk, Jordi. Thanks a lot. But I'm still puzzled by your link between with the endosymbiosis.
Right.
And the reason for that is actually that you start out with a multiplicative growth process in gene length. And so, and from the data that you show in the variance and the mean gene length, I think like, oh, that fits great, that multiplicative growth process.
Mm-hmm, mm-hmm.
So now imagine that we come to the, so it also explains basically why there was such a long time before we reached this critical gene length where, you know, where the protein start to level off. But for me then it's like, okay, this growth process happens and then we reach this critical length. I mean, you say that this transition is actually due to endosymbiosis, but from the data I think like, okay, the growth process just continues with this exponentially growing.
Right, in the site of
So,
genes, correct.
endosymbiosis just came right at the moment that we reached this mean critical
No,
gene
No.
length.
no,
It's like,
no, it's the other way
why
around.
don't I see a break in this growth process there?
Because again, there are two scales. That's the nature of the tension. On one hand, you are totally right. There's a constant gene length, and endosymbiotic doesn't matter at all. If this would not have happened, we would still have seen that constant multiplicative process of gene length from early on to now. Has nothing to do, you're totally right, has nothing to do. Now, it's only when we look at the parallel growth of proteins when something happened. And still without thinking about the endosymbiotic field. Let's imagine it never happened as a mental exercise. So, you have a constant multiplicative process of gene growth. That's the idea of a power law, right?
Yeah.
Scale free. That's the idea of scale free. But you would still find, like early on, there's a parallel increase in protein length, and this is truncated. This is where the tension happened. Why is that? That's our assumption, right? But based on lots of experimental evidence that, you know, longer proteins are not valuable. How you fold a much longer protein, it takes much time and much energy. So there's still no miracle, there's still no biology in here, right? So,
Yeah, so another way of phrasing my question maybe is like, okay, if endosymbiosis would have happened at an earlier or later time, would this critical gene length be different?
no.
I would say yes. Exactly.
That's a good question.
I mean, because this relates to like I understand.
Well, yes.
I understand your point about.
It would be the same because it's of a computational or physical nature, right?
But maybe the search process evolved in a different way, the way they do this new encoding of protein regulation, maybe that regulation completely changes as well. So the algorithm changes at the same time as this thing, maybe that would be different. So given the algorithm, yes.
I think it would have happened at the same time. because that's the physical part, the physical or computational part. The computational part is telling there's a limit, and the limit is here. It's not sooner, it's not later. The limit is here, right? What the physical part is telling, it's only this, that no matter how many replicates of evolution happen in many other planets, if life follows that particular way, that's a limit. It's a physical limit. You cannot get longer proteins, you cannot go much farther in the process of gene regulation based only on proteins. Therefore, there's a limit in the organismal complexity of bacteria. That's all. And you don't need to think about the endosymbiotic theory. But now what happened is that on Earth, in the only evolutionary trajectory of replicate we're aware of, that happened. And when that happened, that unlocked alternative solutions. And as you see in the history of life, evolution, life embraced that opportunity.
Yeah, I understand that.
But I think the prediction of where the threshold is, it's the same regardless of whether the endosymbiotic would have happened before. It could be that then evolution would have had like multiple pathways, right? And the eukaryotic cell or multicellular organism would have certainly started early on, and yet that physical constraint would remain as it is today.
Can I put it slightly different? So would you expect a similar phase transition for the multicellular transition? So this was a eukaryotic development, but there was another major point, which is the Cambrian explosion.
That's a great question.
And so did you actually see
That's a great question. I don't have some solid evidence, but several people, like among others my former PhD supervisor, Ricard Soler, has written kind of perspective papers, more hand-waving that with real data, that would make the point in a very speculative way that all these major transitions in life, origin of life, origin of eukaryotic cell, origin of multicellular organisms, origin of consciousness, could be seen as examples of phase transitions. Now, if you ask my opinion, there's no other major evolutionary transition where you have the same, or people have reached the same level of evidence. Because I think that's the difference between a hand-waving explanation and one with all its faults, with all its limitations, but it's extremely quantitative one and has very, very strong predictive power.
But you need more regulatory.
But you are totally right. Now if you, let's look at, because it's something I've discussed with my colleagues. Oh, we should continue. We had so much fun. It's been so engaging because, again, oftentimes when I talk to students, I emphasize the process rather, because people, oh, it's a paper published in Nature, or in, you have many papers there. That may be okay, but what people miss, and to me what's more interesting is the process and how enriching the process is, right? And because of that, we thought we should continue. And of course, that's a natural thing, go to the multicellular. But to be totally honest, I don't even know what would be the variables there, right? Because here the order parameter, we define that. What would be the order parameter? I have no idea. But I think it's worth thinking. And I propose to my colleagues to start spending some time on that. If anything, to continue the fun. Perhaps we never reach anything meaningful, right? But the same to all of you. I mean, if you are interested in that and you spend some time thinking of that, I would feel very, very fulfilled, because to me that would be the best outcome of my talk, the best measure of possible success of my talk, right? That you or somebody else keep thinking about that. And perhaps in a few years you come out with a paper, I would be very happy. Or even disproving that, right? Because, you know, sometimes it's hard to go, but it's another, Stephen Jay Gould said it, the ability for the great truths and the inspired failures are the two sides of the same coin. So even like when people come up with a paper and say, okay, this algorithmic, it's not correct, the nature of the phase transition is energetic, for example, that would be great, because there was something to that has been pushing the way people think about that, and that could be constraints. It could be that at the end this algorithmic phase transition is bullshit, right? But I think what I really think is that, to me, the key thing is the idea of constraints, and I'm a big believer. And Nick Barton, for example, would have a completely different story. Do I think he is wrong? Not at all. I mean, as always, there's a combination of things. To me, what's very inspiring that they also emphasize constraint, but in their case, the nature of the constraint is energetic. And probably that's much more relevant or much more down to earth or much more easier to explain. I mean, I think he would have done a much better job than I've done if the point was emphasizing constraints are important. He would say, okay, there's a limit to how far prokaryotes can go because there's energy constraints. All life on Earth, on top of having the same genetic code, have the same mechanism of creating energy through a bond in the membranes, through a proton interchange. And therefore it's very easy to see that as the size of the organism increases, as a power of 3 of length, membrane grows as power of 2, relative surface decreases. They would say there's a reason you have higher prokaryotes. You cannot have higher prokaryotes. It's a physical energetic constraint here.
So I looked at, it's the lungfish that has the longest tube.
Okay, which one, sorry?
The lungfish.
Okay, good.
And furs as well.
Good.
So it's 100.
By the way, can I make a tiny random comment? Lungfish is the name of a band, a rock band, which mainly none of you would have heard, that ironically have a song called Creation Story,
Okay.
which is an account of evolution in a song.
But so it has 133 billion base pairs, but most of it's junk.
Yes.
So it's kind of got, it's lost the evolutionary
Well,
switch.
can I stop a
Yeah.
little bit? That's, as people refer to, if you believe something I've been conveying is not junk, it has a huge role, right?
Well, I think there's a lot of
It
repetition.
has a regulatory. It doesn't code for proteins in the sense of, it doesn't code for proteins, but it has a regulatory mechanism.
But these kind of organisms that have these kind of large, in terms of metabolism, it's very inefficient. So they have low metabolism, takes a lot of energy to encode the genome.
Who? Lungfish?
Lungfish.
Okay.
They live in environments which are very stable, so they
Uh-huh.
don't need to be evolutionary kind of dynamic.
Mhm.
So that got me thinking, well, one thing that's a big constraint in all of this search process is what is a viable solution is constrained by the environment, right? So if you do something and then the environment's very dynamic, you need to be able to kind of adapt. So a lot of these species that have these long genomes would have died out in any kind of change in the physical kind of environment of the Earth. So I'm wondering if you look back at this 2,600 million years and you see some sort of big transition in the dynamics of the Earth, right? So there's something, there was the oxygen explosion.
Correct.
So
Correct.
this is maybe why,
Correct.
and that also could be why the cell comes out, right?
You're totally right. As I said, any attempt to be like a theory or an explanation of data, even if it ends up like making very strong it doesn't mean neither it's the only or perhaps the most important one. It just means it's coherent. And many people would make the same point. They would say, well, that's very interesting. I see the value, but what's much more important is that around this time, there was the biggest episode of contamination ever. Photosynthesis, which was a big evolutionary invention, come up with like a toxic product, oxygen. And 85–92% of organisms disappear. That's ironic, right?
So in order to survive, with such
Yeah.
constraints, you need to change the search process, which is why this thing was encoded in the way.
I think that's a great point. I think that's a great point. I've not thought enough about that to give you a minimally interesting answer, but I definitely agree that it's a very, very interesting way of thinking.
Because you wipe out 85% of the things that need to survive, the
True.
old ways don't work. So the algorithm has to change, right? So that's what you see with this explosion thing.
True, true. And yet, the very, very basic, the constraint would remain.
Yeah.
It doesn't matter what's the nature of what triggers this change, but I would say if that's correct, at the very least, should stand that there's a physical constraint and this cannot be changed. Now, what can be different in the interpretation is what's the more relevant mechanism, or perhaps the combination, the suite of mechanisms that allow this finding of new solutions. Right. But again, that goes back to what I said. To me, what's the most beautiful? Reconciling that stochasticity of life with this predictive power of physics, or universality of physics. So the universality would be, we predict there's a constraint. And I think that's very solid. And then the universality would be, how come you come up with a way out? This cannot be predicted by physics. That's the role of biology. And to me, that's what makes it so beautiful, this dialogue. As much as I envy my physics friends, they cannot explain everything.
That's a very good vibe.
There's so much to discuss. Let's end this.